in the natural conditions and also in the glasshouse conditions. That is how we studied the host pathogen interaction studies. Yes.
Okay, thank you.
Thank you, Sunil.
Thank you, sir. Thank you for giving me this opportunity. Yes, sir.
Next is Lakshmi Prasanna.
Yes, sir. Good morning, sir. Can I share my slides, sir?
Yes, please start. Mute, unmute.
Lakshmi Prasanna, are you sharing the slides? Lakshmi Prasanna,
The slides are with organizers. You can share it. She can present from there.
Prasanna,
Actually, she is not online. So there is some. Yeah. Now it's coming. Very good.
Prasanna, come.
Good morning, sir.
Yes, please start.
Good morning to the delegates and professor, my dear other participants. Myself, Ganeshini Lakshmi Prasanna, currently pursuing PhD plant pathology. Coming to the today's occasion, I'm going to present oral presentation on determination of optimum incubation time for the formation and quantification of Xanthomonas axinopodis PVC tree biofilm on abiotic surfaces. Coming to the brief introduction about the disease and causal organism. That is, yes, we all know that acid lime is the fifth most important commercial food crops in India, and there are more than hundreds of biotic and abiotic factors that is causing losses in acid lime production as well as causing economic losses. Among them, Xanthomonas axinopodis PVC tree, which is causing bacterial canker, is the major one, causing economic losses by leading to the export quality reduction. And mostly it is due to the forms of aggressive bacterial strains. Among them, biofilm, which is the major one, which is causing and which is developing aggressive, virulent bacterial strains. And these biofilms are the structural communities of bacteria that which act as a survival ability factors and which protects bacteria from another unfavorable conditions like other bacteria such as antibiotic resistance. So here in this study, we have determined to optimize the incubation time taken for the formation of biofilm as well as the quantification of biofilm, upon which how much the quantity of biofilm have formed on abiotic surfaces like microtiter plates. So we used ELISA micro 96 well microtiter plates reader for the calorimetric quantification of biofilm formed by the Xanthomonas axonopodis citri. And this is the protocol which we have used to calorimetric estimation of biofilms formed by the Xanthomonas axonopodis PV citri. And coming to the results, we have found that among the three different time periods like 24 hours, 48 hours, and 72 hours, we found that 48 hours is the maximum time period to which Xanthomonas axonopodis leads to the formation of highest biofilm compared to the other periods like 24 and 72 hours. And among the 32 isolates, we found that Xanthomonas axonopodis PV citri strain 21 formed the highest biofilm on 48 hours of incubation, followed by Xanthomonas campestris PV citri strain 10 and 6. And coming to among these Xanthomonas axonopodis PV citri 21 strain formed 1.14 micrometers of wavelength at 570 wavelength, and followed by Xanthomonas axonopodis 10, which is 1.4, and followed by Xanthomonas citri 6, that is 1.39. And coming to the conclusion, we after conducting study on ability of Xanthomonas axonopodis forming citri biofilm formation, we found that 48 hours is the maximum time period to form maximum biofilm formation, and the highest was produced by Xanthomonas citri 21 and 10, and followed by 6. And this basic study is useful for this conduction of additional research work on mechanisms involved in in-plantar studies, as well as to create novel techniques to manage the novel biocontrol test, as well as novel target control methods to control this serious disease like cancer. And this information also necessary to identify genes responsible for the biofilm formation, as well as the virulent strains associated with biofilm. Thank you, sir.
Thank you, Lakshmi. I have two queries.
Yes, sir.
If we change the substrate in the medium, the biofilm production will change or not?
Sir, actually, we use nutrient broth as a substrate.
My question is, if I make modifications, manipulations in the medium,
Yes, sir.
whether the production of the biofilm will be changing or not?
It will change, sir.
Okay. Then if it is changing, what is the substrate that increases the more production of biofilms?
In generally, some of the research are saying like host media means host related media will be promoting more biofilm formation by showing apoplastic environmental conditions, sir.
Now, based on this information, in your last slide, you told this can be used for the disease management. How will you correlate it?
For that, we have to study further studies.
What
for
type of-
different
For example, now you have completed your degree program and pursuing the further work.
currently,
What work you will do?
sir, I'm going to study on biofilm inhibitor study, sir, by using nutrient broth as a basic media. But there is a need to study other medias also for correlation of this studies, sir.
Yes, sir. Sridhar sir, others? Participants?
Yeah, one thing, just there was some, only thing, is there any successful example world over using of biofilm inhibitors?
Yes, sir. They're using chitosan at 0.2 ppm in Xanthomonas variegae PV variegae, sir.
In rice?
Yes, sir.
Okay, okay. Thank you. That's all.
Thank you, sir.
Other participant want to ask question? Thank you, Lakshmi.
Thank
Prasanna.
you. Thank you, sir.
Presentation is on the different temperatures effect on growth and life stages of the brinjal shoot and fruit borer, Lucinodus urbonalis, by Mr. Izawul Haq. Is there? He is there? Izahul Haq. I think I am not seeing him in the list. In the
Yeah,
participants list.
in the finally you can call if he's not.
Call him there.
We can go to next one.
One is on the plant parasitic nematodes by Dr. Kishore Kumar. Dr. Kishore Kumar. I think he is there online.
He was
Yeah, one Kishore Kumar is there. Surya.
Can share your presentation. I think he is the lone plant pathologist in the whole presentations.
Okay.
Kishore, are we audible to you?
Dr. Kishore? Yeah,
Can share your presentation.
sir, your voice is very low, sir.
Very
Voice-
feeble. We could hear, but very feeble.
Yes, sir, continue.
Continue, please.
Sir, screen share is okay?
Yes, sir.
Yeah, respected chair, good morning, and the other participants. Myself, Dr. Kishore Kumar, working as a plant pathologist and doing work on nematodes ICR project. And now my topic is bioagents in managing plant parasite nematodes in vegetable crops. That is working on tuber crops, that is elephant foot yarn. Sir, as we know that Andhra Pradesh is the horticulture having a lot of horticulture crops, diversified horticulture crops.
Your voice is not very clear. Dr. Kishore,
It's okay.
you speak very close to the mic so that it will be easy for us.
Sir, now it is okay, sir.
Yes, now it is okay.
Sir, good morning, respected chair and the other participants. Myself, Dr. Kishore Kumar, working at HRS Kovur. And today my topic is bioagents in managing plant parasitic nematodes in vegetable crops. And the topic is in tuber crops, that is elephant foot yam, which have been working at Kovur. As we know that Andhra Pradesh is the horticulture crop, having diversified horticulture crops, both vegetables, fruits, and other spice crops and condiment crops. And within the vegetable crops, I have selected the topic of tuber crops, which I have worked for the last 2 years. And under the vegetable crops, the tuber crops, that is elephant foot yam, which is a major crop being grown in the East and West Godavari districts as one of the major crop, which is used as a consumption directly as a vegetable. So, and then during the surveys being done in Andhra Pradesh as working on the nematodes for the last 4 years, and I found that the nematodes, most of the elephant foot yam tubers are being affected or being rotten with unknown reason. And I have been gone through all the surveys of the different districts wherein that we have found that galls on the tubers. So based on this, we thought of making doing a research experiments. What are the major causes and how to manage this problem, as the vegetables, this tuber is being widely used as a vegetable for the most of the districts in Andhra Pradesh. So with this, we have taken up a research experiments with 6 treatments. Once we found that it is being with the nematodes, as the primary external symptoms is with the galls, which is the characteristics of Melodogena incognita, which has been identified with the Coimbatore Center, that is Melodogena incognita, causing the galls on the elephant foot yam, that is elephant foot yam tuber. And among this, we had come up with the treatments of 6 treatments, that is with cultural operation, that is with application of neem cake, soil application of neem cake along with the seed tuber, And again, next one is microbial consortium, as there is a very good literature being managing the parasitic nematodes with these three bioagents, that is Trichoderma and Pseudomonas and Pislomyces. And the third one is only raising of Indian mustard crop, as we had the literature that the mustard crop is going to be a repellent crop for the nematodes. And we have come up with that another two treatments mixing with both the cultural management, and the other one is microbial consortium. And the local practice where the farmers will be straight away planting the crop, that is tuber crop, as and when they buy from the other farmers, either from West Bengal or Tamil Nadu or from the Kerala. So this is an external view how the nematodes been affected with Elephantium shows the gall, which has been unknown to most of the farmers. They will be taking up with this planting for the new season also, and this is being causing havoc to the new areas also. Unknowingly, this nematode infested tubers, whenever planted in new areas, it will automatically transfer to the new areas. So this has been taken up to the lab, and we found it under the microscope. And this is how the female nematode is been infected, and the egg mass. And this is the egg mass, and this is the female J2, and this is the affected tuber. And here, if you see that the final result, we found that the application of neem cake along with FIM, along with this Trichoderma, along with that again raising the Indian mustard crop up to flowering stage and incorporating in situ has given the highest yield of 45.3 tons per hectare, with the lowest 5.1 of J2 of 200 cc soil, along with 3.3 of 5 grams food tissue. And this is the aerial view, sir. Here you can see how the plant looks from the aerial view, and they treat with tubers treated with that microbial consortium along with raising of Indian mustard crop. And this is how the greenish foliage is there. And if you see here the other light pale yellowish, which has been not treated, this is showing a very clear-cut symptoms how the management of nematodes is being against the parasitic nematodes. And here the water, wherever there is a green luxuriant growth of the plant, and we can clearly infer that this is the impact of how the bioagents are going to manage the nematodes. And as we know that nematodes can be only managed with the integration of both cultural and the biological methods only. And the post of that we can go with the chemicals also. But here the problem is the chemicals as such that is not a clear cut targeting the nematode. So we have to recommend to the farmers of both using the cultural and the biological method. And this is how the symptoms, this is how the treatment results. If you see here the treated, this is the result control plot where the tubers are totally malformed or misshaped with lot of galls here and there. If you see that the integration of this treatment 5, which is with good shape of the tubers, which is very good, where the farmers can get high yield also and high price. So with this, our recommendation is that soil application of neem cake along with FIM, and again treated seed tuber treatment going for planting, and raising of Indian mustard crop up to flowering stage and incorporating in situ has recorded the lowest nematode population and with the highest yield of the 45.3 tons per hectare. And apart from the Elephantium, we have found that other Colocasia also being affected with the nematodes, which has been there. We can find the galls. These galls are the first primary external symptoms of the incidence of the parasitic nematodes. Finally, as nematodes is nowadays becoming a threat to most of the horticulture crops, especially because of continuous supply of fertilizers and nutrients, along with that lot of care, and moreover there is no dry period also, continuously raising the crops continuously with which are the major favorable factors for more development of the nematodes. So unless until we go with the cultural management practices along with biological, then only we can effectively manage the parasitic nematodes. As a prophylaxis, we have to be taken up the utmost care, and with this we can go and manage all the incidents, and there so that the farming community can be most helpful. And with this I conclude my topic today, that is the bioagents in managing the plant parasitic nematodes. Thank you for giving me this opportunity.
Thank you, Kishore. I have few queries. Is it Gajendra variety what you have used for
Yes,
your
sir.
study?
Gajendra, sir.
Then yesterday Dr. Sridhar mentioned for the entomofungal pathogens, you have to mention the CFU, what was applied at the time of application, what was the CFU. Then while recording the observations, what is the CFU of the bioagents. Yesterday he mentioned for the entomofungal pathogens are the another presentation. So that will give the value for your presentation. CFU count of the bioagents you have to give. Then you mentioned about you are using the Indian mustard for the as a repellent.
Correct, sir.
Isn't it? You are in the AICRP for tuber crops?
Yes, sir.
There is one more, this Sri Bhadra on sweet potato variety is there. Sri Bhadra S1010 selection. That is used as a nematode trapping crop. That also you can try. One more is the marigold.
Okay, sir.
So in your next day experiments, this is suggestion from my side. Sri Bhadra sweet potato and marigold you try that are in the crop rotation or in the Sri Bhadra as the trap crop. That also gives a better result.
Sure, sir.
And followed by the nematode damage, there are many storage rods come in the elephant foot yam, especially.
Correct, sir.
So when you are using these bioagents, whether there is reduction in those fungal in storage rods also, that will be adding value to your work.
Sir, actually regarding storage rods, I had that observation only for preliminary observation, sir, that there is a storage life of another 45 days. The nematode health tubers were not being affected with any other storage rods, sir. Even at the time of harvesting also, the healthy tubers were kept like that for storage. Why? Because we sell the tubers also for the farmers for the new season. And just primary found that the tubers are very much bigger without any rotting also, sir. But that experiment, as you said, sir, we'll take up in a research way also to find out how best it is going to be helpful in the storing of the tubers also.
Because these tubers are saved for as a seed tubers up to 6 months. There, this storage rods are problem. These bioagents will help.
Correct, sir.
Sridhar sir.
Yeah, okay. Here generally, whether this Trichoderma viride and Trichoderma herzianum, what is your
Parallel,
next
sir.
Sir, it
observation?
is Herzianum only, sir. We have used Herzianum only.
But you have
But
written
you
as
mentioned
viride.
viride, whether viride is existing or what is the position of viride?
Sir, actually, as per the availability, we have taken up with Herzianum, sir. Viride, there is a lot of controversial, but by observing the spores and all, even I am also not that clear that whether it's Herzianum or Viride, sir.
Yeah, you have used viride. That's why it should be like herzianum. Another one is you total treatments in your experiment are 6 na. How many replications you use?
तो डीन आएंगे और वो हटा गया हूँ
Hello.
3 replications,
क्या
sir.
रहता हूँ
If it is 3, it is not sufficient for this analysis on our RBD. It comes to only degrees of freedom of 10. It is not sufficient for analysis. Just please look into that.
Okay, sir. Okay.
Okay. Okay. Right.
Thank you, sir. Thank you so much.
Right. Right.
You, Dr. Kishore.
Sir, yeah, please, sir.
Thank you. Thank you.
Okay, sir. Thank you so much.
Any other questions? Any other participants? If no other question, we will go to the next presentation by Mr. P. Naveen Kumar, Disease Management of Collar Rot in Elephant Foot Yam.
Naveen?
Yes, ma'am. Yes, ma'am. Madam, is it visible? Hello.
Yes, it is visible.
Yes, sir. Good morning to all the dignitaries. Myself, Naveen Kumar. I am pursuing MSc Plant Pathology in College of Horticulture, Venkatramanagodam. Today, I am here to present my research topic, that is disease management of collar rot in elephant foot yam. Coming to introduction of the crop. Elephant foot yam scientific name is Amorphophilus pinifolius. It belongs to the family Araceae and it is referred to as king of tuber crops due to its high yielding potential and culinary properties and medicinal utility. Elephant foot yam rich in carbohydrates, minerals, vitamin A and vitamin B. But in elephant foot yam cultivation, the major problem is instance of
Voice is not audible. Mr. Praveen, your voice is not audible. Naveen, you are not audible. Organizers, are you able to listen to him?
No, I think he is not audible.
Hello, Naveen? Naveen,
Madam,
you are not audible.
sir, madam, little bit network problem.
Yeah. Now you continue.
Now you can
Continue.
Okay, ma'am. The disease normally occurs during the peak period of the rainy season. That is mainly in the months of August and September. Hence, we conduct an in vitro experiment to know the best method to control the collar rot of the elephantiasis. These are the symptoms of the plants in the regions of West Godavari. Coming to materials and methods we used in this experiment are mancozeb, copper oxychloride, thiophanate methyl, propiconazole, azoxystrobin, flucelazole, tebuconazole, difenoconazole, carbendazim plus mancozeb, carboxyl plus thiram, and treatment level as control. And we used these fungicides in 3 replications, that is 500 ppm, 1000 ppm, and 1500 ppm. In case of contract fungicides, we used 1500 ppm, 2000 ppm, and 2500 ppm. Coming to bioagents, we used Trichoderma viridae, Trichoderma haggianum, Trichoderma raceyi, Pseudomonas fluorescens, and Bacillus subtilis, and T6 as control. And the replications of 4 using design of experiment is CRD. Coming to the results and discussions, Trichoderma viridae inhibits the mycelial growth, that is 60.82%. Trichoderma haggianum 76.11%. Trichoderma raceyi 54.94%. Pseudomonas fluorescens 83%, and Bacillus subtilis 87%. Among these, Bacillus subtilis majorly inhibits the mycelial growth. And coming to contact fungicides, that is mancozeb and copper oxychloride. Mancozeb inhibits the mycelial growth completely at 2500 ppm concentration. This is the picture that is shows the antagonism of various bioagents against Sclerotium dolce. Here is contact fungicides that mancozeb completely inhibits the mycelial growth at 2500 ppm concentration. But copper oxychloride shows no inhibition of the mycelial growth at all 3 concentrations. Coming to the contact systemic fungicides evaluation, we used 3 concentrations, that is 500 ppm, 1000 ppm, and 1500 ppm. In this, azoxystrobin, propiconazole, flucelazole, tebuconazole, and Vitavax completely inhibits the mycelial growth at all 3 concentrations. And SAF, that is carbendazim plus mancozeb, inhibits the mycelial growth at 91% at 1500 ppm concentration. These pictures shows the inhibition of the mycelial growth of various chemicals. Come to conclusion. So among all the tested chemicals, 5 fungicides that controls the mycelial growth 100% at all 3 concentrations. So based on the cost ratio, we recommended the propiconazole for the control of collar rot disease. After that, carboxyl plus thiram and tebuconazole, flucelazole, and azoxystrobin is used as alternative for the controlling of the collar rot. In case of bioagents, we recommended Bacillus. Sir? Continue. Yes, sir. In case of bioagents, we recommended Bacillus subtilis subtilation for the control of the collar rot. In alternative for that, Pseudomonas fluorescens and Trichoderma haggianum is also used for the control of the collar. Thank you so much for giving me this opportunity.
Thank you, Naveen.
Sir.
Naveen, this inhibition of Sclerotium dolce with the fungicides and bioagents in vitro has been reported n number of times. I would have loved to see some in vivo experiments, because already there are so many reports, especially in the elephant footium itself. Many people have already carried out many work.
Yes, sir.
You should have started with in vivo experiments. And one more information: there is no viridae; it is aspergillum. Viridae produces the difference between the Aspergillum and Viridae is the finely accumulated conidia is produced in the Aspergillum, and in the Viridae it is conspicuously bigger spines will be there. Viridae will not grow at 35 degrees centigrade. Aspergillum will grow, and Aspergillum will produce chlamydospores, and Viridae will not produce. And by wrongly we were using the Viridae, but it has been changed in 1996 only, and we also published in 2013. So before using the names, as Sridhar sir in the previous presentation already told, you cross-check that one, and you wanted to tell something, you tell.
No, sir. Sir, we are conducting experiment under pot culture, sir. This is one of the objective.
But I would have loved to see the pot culture or the field experiment, because this is very well known. Trichoderma or Pseudomonas bacillus inhibiting this Chlorotium in vitro, or these fungicides inhibiting is very well known.
Yes, sir. Yes.
The evaluation, at least you should have used the different combination products.
Yes, sir. Yes, sir.
Sridhar sir. Sridhar
Okay.
sir.
Okay, yeah, in your Naveen, in your experiments, you used so many triazoles. What is
Yes,
the
sir.
general mode of action of triazole fungicides?
Those are fungistatic, sir.
How they will make the fungus static?
Understand.
Difference between fungistatic and the fungicide.
Sir.
What is the difference?
It means growth inhibition, you mean to say?
Yes, sir. Percentage of growth inhibition, mycelial growth inhibition.
Yeah, how it happens. Actually, some mode of action will be there. Cell wall development stops, something like ergosterol inhibition, something.
Yes, sir. Yes.
All fungicides have some specific mode of action. When you are using, it is better to know how they will work.
Yes, sir.
Okay, thank you.
Thank you, sir.
Shall we go to next presentation, sir?
Yeah, next will go. Yeah.
Sushma, first report of gummy stem blight in Ridge God. I mean, you can share. Please unshare the screen.
Good morning, sir.
Yes, please. You can start.
A very good morning and a warm welcome to one and all. Myself, Sushma, PhD scholar from Dr. YSL Horticultural University. Today, I am here to deliver my oral presentation entitled on first report of gummy stem blight of Ridge God caused by Didymella species in Andhra Pradesh. Ridge God is an important vegetable crop used widely in our day-to-day life in cooking, and it belongs to the family Cucurbitaceae, bearing a chromosome number of 2n is equals to 26. Despite of its nutritional value, it was rapidly infected by plethora of economically important diseases caused by many fungi, bacteria, and viruses. Among them, more recently, a complex malady named gummy stem blight, which might have also present earlier, but it is increasing in its severity and intensity and causing serious threat for the production of cucurbits in India. So this sounds to be a need of art for the plant pathologists to deal with. And considering this, the present investigation was carried out with the following objectives: to conduct survey to know the disease incidence in Kurnool and Kadapa districts of Andhra Pradesh, and to characterize the pathogen morphologically and molecularly. So during the survey, the visual ratings on the leaf were scored by using the scale given by Zhang and his co-workers, and the percent disease index was calculated by using the formula given by Wheeler. So during the survey, symptoms were observed on all the aerial parts of the plant, right from the seedling to the harvesting stage. On the leaf, initially we have observed we have a leaf spot, and majority of the leaf spot were present on the leaf margins. And as the time passes, these spots will be coalescing and enlarge its size and turn into dry. And rapidly the tissue necrosis started from the leaf margins and progressed towards the base of the leaf. And when we look at the symptoms of the stem, the water-soaked spots initially occurred at the joints and then enlarged into the streaks and then turned into the creamish colored cankers. And the stem also splitted longitudinally and showed the golden amber colored gummy exudates. So might be the reason this disease was named as a gummy stem blight. And after this, the gummy exudates will be developing into the hard encrustations. When we look on the symptoms on the petiole, we found that petiole is deeply wrinkled and it is showing a drooping appearance. Under severe infestation, we have also found symptoms on the fruit, and symptoms were found on the fruit at the blossom end of the fruit, showing a black rotting-like appearance. And we have also found the gummy hard droplets upon the rotted tissue. So the symptoms, whether they are present on the leaf, on the stem, or on the petiole, or on the fruit, we found abundant tiny studded dot-like structures that we can call them as fruiting bodies, like pycnidia and the pseudothecia, which is the chief diagnostic symptom for the characterization of this disease. And then we collected the infected samples, and during the survey, the percent disease index ranged from 8.5 to 46%, and the mean percent disease index was observed to be 20 and 13.42 in YSR, Kadapa, and Kurnool district respectively. And we found the more percent disease index in YSR, Kadapa district. Might be the reason, many reasons might be there, and so it might be due to the favorable environmental conditions like high relative humidity and continuous cropping of the cucurbits, and it might be fill up the faster development and the advancement of the disease. So we have brought the infected tissues to the laboratory and conducted the molecular characterization, and the pathogen was isolated on potato dextrose agar medium, and we found that pathogen growth with a dirty white to olivaceous green in color, and after a few days it turned to gray, and after it turned to finally black in color. When we see the reverse position of the Petri plate, the growth of the mycelium was observed to be radiating from the center in the concentric rings. So the microscopic studies revealed that the mycelium of the pathogen was septate and irregularly branched, and we also found small black globular pycnidia and unending stream of pycnidiospores, which are cylindrical in shape with rounded ends, and most of the pycnidiospores were mostly monoseptate and they are rarely non-septate. And we also found the pseudothecia and bitunicate assay, and ascospores were also found, which are having the rounded ends. These are the hyaline, and we also found the slight constriction at the septum, which is the diagnosis feature. We can differentiate it from the pycnidiospores. And for confirming the pathogen at the molecular level, we have isolated the DNA by following the CTAB method and conducted the PCR by the given conditions, and the PCR products are subjected to electrophoresis, and we have conducted the PCR by using the universal primers like ITS1 and 4, and the PCR product was amplified at the band ranging at 545 per, and they were sent for sequencing, and the results showed that it was showing much homology with the Didymella species, and we concluded that the cause of the disease is Didymella species. And looking at the future, as it is the only the preliminary work done, and more focus to be done on the management aspects to fulfill the and to safeguard the livelihood of the cucurbit farmers. Thank you very much for giving this opportunity.
Thank you, Sushma. Sushma, I have a few queries.
Yes,
You
sir.
mentioned about a Didymella species,
Yes, sir.
whether it is Didymella brioni or some other species.
Sir, it was only characterized up to the species levels, genus level, sir. Species
Okay,
level.
What is the new name for this Didymella brioni? Any synonyms, new names?
Yes, sir. It is also called as Stagnosporosis cucurbitacearum or Phoma cucurbitacearum, sir.
What you could have done, you have done the amplification, that sequence. When you have checked with the NCB, it is found very similar to the Didimella, isn't it?
Yes, sir.
Further, for further identification, what is the next step? What we can do?
Sir,
Next step I want to confirm. Now genus level you have confirmed. I want to go to the species level. How to confirm further?
by using the species specific markers like scar markers, we can identify up to the species levels.
Any other method? Any other method is there to identify, to discriminate? See, now you have Didimella from different places you are bringing. Watermelon, ridgegod, and other cucurbits. Now you want to further
Yes.
narrow down for the ridgegod crop. How will you do? Among the different isolates of Didimella, multi-gene phylogeny you should do. If you
Yes.
are continuing this work, you try to do multi-gene phylogeny, so that will differentiate whether it is very close to Brioni or some other species.
स्टार्ट वीडियो आ गया ना, अभी तो चल रहा है ना। और अपना प्रोजेक्ट कहाँ से शेयर करें? ये शेयर
Yes, sir.
Hello. Yes, so this is now it is an increasing disease in different cucurbits. What is the present farmers' practice for managing this? What difficulties they are facing?
Sir,
And whether any chemicals are responding to this in suppression of this disease, or still no chemical is working? What is the farmers' opinion?
sir, as it is a emerging disease now, much work has not been done, sir. So only up to the characterization of the pathogen.
Pyroclostrobin group is okay, sir. So far.
Can be suggested.
Yeah, okay, thank you.
Any other participants having any queries for Sushma? Okay, thank you, Sushma. We'll go to the next presentation.
Thank you, sir.
P. Rajeshwari, behavioral responses of Nephopteris. I don't see her name here, no? Rajeshwari is there?
Yes, sir, she's here.
Present there.
Can I start, sir?
Yes, please.
Yeah, please go ahead. Share the slides. Proceed.
Good morning to respected chairman, Dr. Sriram sir, and co-chairman, Dr. V. Sridhar sir, and other dignitaries and all the faculty members. A warm welcome to my presentation. I'm G. Rajeshwari, working as assistant professor at College of Horticulture, Anantarajpeta. Today, I'm here to present a topic on behavioral response of Nephopteryx eugraphila to host plant volatiles. Coming to the introduction, the host plant volatiles are nothing but called as
Slides, sir. Please share the slides.
Yes, sir.
Visible or not?
Sir, is it visible now?
Yes. You can start. Please clear.
So the plant volatiles are nothing but called as chiromonal cues, which are very important. They play a major role in host recognition of insects through chemical signals. And this Nephopteryx species being a major pest of Sapota, the management practices are very difficult due to the webbing pattern of the this insect, especially if we spray any chemicals. the chemical management is very difficult because of the webbing pattern of this insect. The grown-up caterpillars, they start secreting the silken threads to make a web, and they stays inside and feed. So in this context, the better option is the using of semiochemicals could be a better option to manage this notorious pest. We have studied the influence of plant volatiles to identify the host location of this Nephopteryx eugraphila species. It's not changing. No.
It's moving.
So firstly, we have collected the plant volatiles of both infested as well as uninfested plant volatiles through air entrainment system. In air entrainment system, the foliage, some of the leaves of support leaves, both infested and uninfested leaves are covered with a polythene cover, and 2 Poropack tubes were inserted into the polythene cover, and that will be tied with a tape. And 1 tube is to pass continuously the activated charcoal filter air, and another tube is to trap the eluted volatiles in the form of fumes. And this was kept for 24 hours. After 24 hours, we eluted the entrapped volatiles by using different solvents like diethyl ether or hexane. So here in this study, we have used diethyl ether as a solvent to entrap the, eluted the volatiles. And further, we went for behavioral studies by using white tube olfactometer, and a 10 microliter of volatile sample was offered on a filter paper. Then the insect was released from this point, and each sample was tested for 30 insects, and the results were showing like in single choice essay, the female moths of this Weber moth preferred plant volatiles when compared to the control. But in case of dual choice essay, the infested volatiles attracted more moths than healthy plant volatiles. Then further, we went for identification of compounds through GC-EAD and GC-MS. This is the GC-EAD, gas chromatography, which is coupled with electroantennogram detection, where an insect antenna is dissected and placed on an electrode. Simultaneously, 2 microliter of a sample volatile was injected to GC to check the response of moths. And the EAD peaks was, the responded chemicals was identified by using GC-MS, that is gas chromatography, which is coupled with mass spectrometry. So this is the GC-EAD trace, the antennal response of female moths to support a healthy plant volatiles. The number is nothing but the chemical compounds which are denoted here. And a total of 9 chemical compounds were eluted response to female moths. And this is the gas chromatography, a picture of support a healthy plant volatiles. And this is the antennal response of this female moths to infested volatiles. Here also a total of 12 chemical compounds were elicited response to the female moths. This is a gas chromatogram picture of volatile profile of infested web volatiles. And after that, after GC-EAD and GC-MS analysis, the GC-EAD active compounds, whatever the available compounds are, available in the market, those were purchased from Sigma Aldrich. And again, we went for behavioral studies for further confirmation. A total of 8 chemicals were tested individually in the laboratory. And among the 8 chemicals, only 2 chemicals, those are P-cymene and methyl salicylate. These 2 chemicals have shown significant response towards the attraction to the female moths. And further, the chemical compounds which showed significant response, those 2 chemicals was used to prepare a synthetic blend, and again tested in behavioral studies by using a white tube olfactometer. The synthetic blend in the first single choice essays, the synthetic blend attracted more female moths when compared to control. Again, synthetic blend was checked with healthy plant natural volatile. There also we have seen the moth preference to synthetic blend. But when we compared it, when we tested along with synthetic blend and infested web volatile, here there was no significant difference was observed, but the moths were attracted more to synthetic blend. So from this study, I could conclude my work by saying that the female moths of this Weber was attracted to plant volatiles, and also the chiromonal compounds which are identified in this study, those are P-cymene and methyl salicylate, they have shown a significant response.
In case of synthetic blend also, they have attracted more moths. But when compared to the natural host plant volatiles, it attracted both, means the moths were attracted both to synthetic blend as well as natural host plant volatiles. So the semiochemicals could play a major role in host recognition of this insect. And also we can, up to lab studies I have done, sir, and we can go for changing the lure composition. I mean that P-Simon and also methyl salicylate, the composition can be changed by making some alterations in the lure composition. We can go for field studies also. So further field studies need to be carried out, sir. Thank you for this opportunity.
Thank you. Sir, Sridhar sir.
Thank you. Regarding this study, you have already done lab studies, you told. Just
Yes,
one preliminary. First question will be, how do you relate sapota with ethnic vegetables?
No, sir. As a horticultural crop, I presented here, sir.
As a horticulture, okay, but
Because the
this
theme
we
is
are going.
Sir, this theme is pest and disease management, not like particular ethnic vegetables.
No, this first time I'm seeing a fruit crop here. This
Yes.
conference is on ethnic vegetables. I had a doubt. Suppose there are few fruits which are used as vegetables also. Is there any incidents where sapota is eaten as a vegetable? That's why I was asking.
Sir,
And
no, sir.
I think regarding blends, what is the success rate? How much, suppose if you are releasing in a Y2, some 10 females you are releasing, how many are getting attracted to that? To the
Not
blend, right?
sir. For each sample, we have tested 30 insects, sir. More than 18 to 19 insects were attracted to synthetic blend. So out of 30.
Out of 30, okay. This, what is the origin of this sapota?
Mexico.
Yeah, somewhere Mexico. Majority will be in Mexico, South America. It is not our, not even Asian also. Then what is the follow-up of your work done?
Sir?
Follow-up of your work, this one. Anything is being happening in continuation to your studies?
Yes, sir. We need to conduct in field trials also need to be carried out, sir.
Yeah.
Further, by
Yeah.
using this chiromonal use, we can do some field experiments also.
Yeah. What is the basis? Something, some alamone and chiramon. Can you relate them here? Alamone and chiramon.
So chiromones is a specific chemical compound which is released by a particular organism, and that can change or alter the behavior of the receiving organism of the different species, sir. For example, plant, if a plant releases some chemical compounds, so that will also attract pests as well as natural enemies, sir.
Okay. Which is beneficial for the releaser, alamone or chiramon?
For releaser, alamone is beneficial, sir. For chiramon, it is harm to a releaser. It is beneficial to receivers.
Receive, right. Okay. Dr. Sriram, you just.
Thank you, madam. I have only one query. Is there any other semiochemical based management of any other Weber, leaf Weber?
Sir, in case of Webbers, these Webbers belongs to the family Pyralidae. In case of this Pyralidae family, nowhere the works was carried out till now, sir. For my PhD, I worked on mango leaf Webber, sir. That also up to field lab studies I have done, sir. Field studies need to be carried out. So at the during my yes.
What's the major compound that you identify that will be useful?
So in this study, P-Simon and methyl salicylates, they both contributing more attraction towards the insects. This Weber moth.
Any other question?
Anybody else?
Thank you.
Okay, thank you.
Thank you, sir.
We have one more presentation by Jemima. Her name
Yes,
has
sir.
been included.
Yes, sir. Good morning, sir.
Have the presentation.
Yes, sir.
Others can be muted.
Sir, am I audible, sir? And is my presentation visible, sir?
You are audible. Your presentation is getting loaded.
Yes, sir.
Yes, it has come.
Yes, sir. Shall I start, sir?
Yes, yes.
A very good morning to one and all, respected chairperson, co-chair, and all the delegates and dignitaries. I welcome you all to my presentation. Myself, Dr. N. Jemima, I am from PJT SIU, and I'm here to present my part of my PhD research on studies on dissipation and persistence of dimethiate in cauliflower. Cauliflower is one of the winter vegetable crops which is rich in vitamins, dietary fiber, and minerals, but it is affected by various abiotic and biotic stresses. It is reported that about 60 to 80% of yield losses occur due to various insect pests in cauliflower. So here are some of the major insect pests. And apart from lepidopteran pests, we have the sucking pests, which act as vectors for viral disease. And apart from that, they suck the sap and result in qualitative and quantitative losses. Generally, farmers are going for spraying of various insecticides. Dimethoate is one of the cheapest and easily available and effective insecticide being used in cauliflower. So, but for any pest management, selection of insecticide and timely spraying and proper dosage is very important, because the indiscriminate sprayings and calendar-based sprayings are leading to presence of residues in the produce, apart from leading to resistance development and resurgence. So there is always a need to minimize the dietary exposure to the pesticide residues, and hence there is a need to go for safe waiting periods. And also, in case there are any residues at in the harvested produce, there is every need for taking up various decontamination methods. So with this objective, we have taken up this study, and the null hypothesis is that the residues, the cauliflower is free from residues of dimethoate at harvest. So this trial was taken up at Farm College of Agriculture, Rajendra Nagar. The cultivar Davar was transplanted 1 month after sowing during Rabi 2019-20. So dimethoate, it was sprayed twice at 15 days interval during curd initiation stage. And after the second spray, we have collected the cauliflower curds at a regular interval of 0, 1, 3, 5, 7, 10, 15, 20, and 25 days. And we have followed Quetches method, which is quick, easy, cheap, efficient, rugged, and safe method for extraction of dimethoate residues. And we have analyzed the residues on LC-MS. This is the schematic representation of the sample collection, homogenization, centrifugation, and after all the chemical processing, the 1 ml supernatant has been injected into LC-MS, and the residues have been analyzed on LC-MS. So prior to the quantification of the results, we have quantified the method, that is validated the method as per SANTE 2017 guidelines, and the linearity and fortification studies were well satisfied as per the SANTE rules. Coming to the results, we can see that at 0 day, that is 2 hours after spray and 1 day after spray, the dimethoate residues in cauliflower were relatively higher, that is 5.13 to mg per kg and 4.341 mg per kg in 1 day after spray. And these residues have dissipated rapidly by 3rd day, reducing up to 82.95%. And by 7th day, the residues were 0.150. And by the 10th day, the residues reached below the limit of quantitation of 0.05 mg per kg. And after 10th and 15th day, almost there were no residues found in the cauliflower samples collected. So from these values, we have arrived, we have plotted a curve wherein we have obtained the regression equation, and from this regression equation we have calculated the half-life. So the half-life of dimethoate in cauliflower was found to be 1.23 days, and based on the available FSSI MRL of 2 mg per kg of dimethoate, we have gone for calculation of waiting period. So the waiting period, safe waiting period after dimethoate spray was found to be 1.79 days. So in conclusion, we can see here the graph shows how the dimethoate residues, which is depicted in red here at 0 and 1 day, which were higher, rapidly dissipated to more than 80% by 3rd day, more than 95% by 7th day, and by 10th day, it was almost dissipated to less than LOQ. And based on the FSSAI value, around 2 days is the safe waiting period after spraying of dimethoate in cauliflower. So to enjoy safe produce or for the consumption, so there is every need to maintain not only the correct insecticide, the correct dosage, but also safe waiting period, so that the produce is free from pesticide residues. So in conclusion, I want to say that not only this crop, any vegetable crop, there is every need to follow the safe waiting period. This study is a part of my PhD research, and I'm very thankful to my university, Professor Jaishankar, Telangana Agriculture University, and my guide, Dr. G. Sridevi Madam, and my committee members, and my team at Pesticide Residue Laboratories. Thank you one and all for giving me this opportunity.
Dr. Jemima. Sir, Sridhar sir, please.
Yeah. Okay. This one, what is the dose you have used? 200 grams AA per hectare? Suppose,
yes, sir, it is. Yes, sir. It is coming. Yes, sir.
Per liter, how much you are using?
It is coming to 1.6 ml per liter, sir. Based on the calculation. Roughly 2 ml per.
Yeah, dimethoate is already registered chemical for cabbage, I think. What is the purpose of taking for cauliflower separately? That studies was not done earlier.
Yes, sir. Studies were done in various parts of the country, but then again, since the dispersion pattern also depends on the temperature and region, sir, in Telangana, and so that's why we have taken up this study, sir. And also, it was actually dimethoate was taken as a control check, sir, chemical. So, but when we got aphids, we found other than other chemicals, dimethoate was better in controlling aphids. So we have gone for residue studies as well, sir.
Now farmers mainly be using this dimethoate only, cabbage, cauliflower. There are so many eco-friendly molecules have come now, which are recommended. Even spinosad at lower dose has come with some companies, 2.5 SC formulation. And here, one more thing is, here you have given sprays at in the initiations now for that flower. That one, suppose this is same 2 days waiting period, whether it is valid just before 2 days before harvest, can it be possible?
No, sir. Again, when the curd is developing, when the size is developing, sir, so the spray matrix, more of the spray will fall on the curd, sir. So when the curd is fully developed, there is every chance of retaining the spray fluid, sir, chemical.
That is true. In early stage, that is fine. It will dissipate, and because of growth of that curd, dilution factor may be there. And then what will be the recommendation from your study to the farmers?
So mostly our recommendation is not to go for any spray just 1 week before harvest, sir. Minimum they should follow at least 7 days of waiting period, sir. Because till 7 days
being a systemic, we should tell minimum 1 week that we should tell.
Yes, sir.
Okay. Okay. Thank you.
Thank you, sir.
Thank you, sir. Thank you, Dr. Jemima. Sir, one more presentation is pending, Dr. Haq.
Yeah, you can. Yeah, we will call him. Yeah.
He's not down.
If he is not there, shall we proceed to
Yeah,
poster presentation.
posters. Okay, okay.
Because we have 9 presentations. Because
9 into
3 plus
okay.
2, madam. Moderator has to.
3 plus
2
2.
plus 2 or 3 plus 2?
3 plus 2.
3 plus 2. Okay.
Because they have a plenary session, they have to get ready for that.
Okay, okay. That will.
Make it fast. First presenter is
Sai
Sai
Meghna is
Meghna. Sai Meghna is not there.
yes, sir. We call for the second.
Second
Naveen.
is G. Naveen.
Yes.
Sir,
Yes, please.
sir,
Yes,
Share your
Naveen.
presentation.
sir, sharing. Sir, and can you visible, sir, poster?
Yeah, you have to open your file. What you could see is you have not yet opened the file.
Share the screen where that folder is visible.
Or initial, you first open the slide, PPT slide, then you share. That will be easy.
Shall we go with the next presentation? You can get it.
And they are doing from that side.
Naveen, we are sharing the slide.
Sir,
Continue.
Yeah, please go ahead.
sir, good morning, sir. Today I am here to present my poster, sir, on the topic of significance of images for effective pest management in vegetable crops, sir. These image stages finding food. These image stages to find food quality and environment to increase their species success, sir. These emissions can move from distance ranging from few hundred meters to 100 meters. The most active immature stage to cause this damage for crop is larval stage, sir. Objective to study these immature stages of insects as pest crops and as natural enemies. Significance of these natural enemies are some of the natural enemies like ladybird beetle and grace leaf wings, and these are can use as natural enemies. To these, one ladybird beetle can eat 50 aphids per day, sir, as an adult. These grace lacewings larvae can eat up to 200 aphids per week. The Cryptolaemus montessori as grub can consume 900 to 1500 millibugs in its development. The major pest of cruciferous vegetable is diamond black moth, sir. This been controlled by larval parasite Plutella, Cotacea plutella. In potato crops, the major pest is potato tuber moth, sir. These can control by egg larval parasite Chilonis black bunny. Significance of crop matches. The crop pests are Aplanthum beetle grubs, sir, and grubs and nymphal stages and larval stages, grub stages. These are the major image stage to cause pest for the crop. This plasma beetle grub feeds superficially on the leaf surface of leaf. This can cause estimated to cause 10 to 15% in damage in normal years and 20 to 30% in heavy infestation. The another image stage, nymphal stage in thrips can cause up to 20 to 100% of damage on the flowering of cowpea, flowers of cowpea. Root grubs, other image stages are root grubs can feel rootlets, while these can cause 40 to 50% yield reduction in coca, sir. The conclusion, so these image stages can also used as predator to control the crop pest in vegetables and other crops, and these can form as predators as a lot of significance in pest management. And other image stages like eggs and pupal are not feeding stages, sir. Thank you, sir.
Yes, sir.
Yeah, you mentioned Trichogramma chilonis, one of the egg parasite. Can you connect this with any of the few these ethnic vegetables where it can be used?
Okra, sir.
Since yesterday we are discussing where this Trichogramma chilonis can be recommended, because commercially also it is available. What are the crops you can use this particular egg parasite?
In okra crop, sir.
What else? In okra, against what pests?
In fruit and shoot borer, sir.
Okay, next. So that you will recollect whatever ethnic vegetables it can be useful.
Cabbage,
Brinjal.
sir. Brinjal.
And brinjal against?
Same, sir. Fruit and shoot borer.
That is where it is already a recommendation is there, clear-cut recommendation. It can be used also.
Yes, sir.
What are the other commercially available biocontrol agents in India?
Sir.
What are the other commercially available biocontrol agents in India?
Sorry.
Which institute is working exclusively mainly on biocontrol agents? Whatever you mentioned, they're all having some practical utilities. That is the reason I'm asking. When you are giving this, how immediate thing is farmers will ask, yes, you are telling this should be used. Where I can get? That is the next question. We should be able to get ready with that answer. Okay?
Okay.
Okay.
I have only 2 queries. Where did you get these photos? Whether photos were taken by you or downloaded?
Sir, download.
Where all these photos were taken by you?
Yes, he is telling, sir, he is agreeing. Downloaded only.
Then most of the genus names you have not given the capital letters.
Capital letters, yes.
Or the Kelonus. First letter it should be capital. No?
Yes, sir.
Whenever you make presentation, take care of while writing the scientific names. Potatia, it is C is not capital. Kelonus blackburni C. See, some importance should be given for presentation.
Okay, sir.
Sir, shall we go to next one, sir?
Oh, next one.
Okay. Dr. Y. Kavya.
Good morning, sir.
Please. Naveen, you can unshare the screen.
Madam, please share my presentation so that I will continue, ma'am.
Yeah, we are sharing.
Sir, shall I start, sir?
Yes.
Good morning to the chairman, co-chairman, rapporteurs, and the moderator. Myself, Dr. Y. Kavya, scientist at Horticulture Research Station, LAM. Now I am going to give a brief topic related to advances in PGPR-driven soil health management. And coming to the PGPR or rhizobacteria or plant growth promoting rhizobacteria, normally we will be calling them as PGPRs, and the term was given by Joseph Kloepfer. And these are the free living bacteria or some other organisms that are able to colonize the rhizosphere, and these are mainly helpful for the plant growth and improvement of the soil health. And coming to the main characteristics, these are able to colonize the root surface and compete with the other microbiota, and finally helping in the plant growth. And what are the main classification are? These are useful as biofertilizers, phytostimulators, rhizoremediators, biopesticides, etc. So how are they reflecting the soil health? These are able to build up the soil fertility and the soil structure. And similarly, they are able to compete with other harmful organisms and finally helping in the plant growth. And what are the perspectives are? They are able to secrete some of the auxins, gibberellins, indolactic acid. In such, they are able to stimulate the plant growth. Similarly, by bioprotection, some of the organisms like Bacillus stimulus, and they are able to secrete some of the enzymes and helpful for the protection of the plants by restricting the activity of harmful organisms. Similarly, they are able to microbial degradation, like they are able to degrade some of the pesticides, some of the xenobiotics. And what are the recent advances are some not only individual microbial organism used as PGPR, some consortial effective microorganisms like formulation, coming to biocapsules like formulations, and biofertilizer efficiency enhancer. These are some of the recent advances in this PGPR activity. So coming to the effective microorganism, these effective microorganism, this is a group of 3 different types of organisms like lactic acid bacteria, photosynthetic bacteria, and some yeast. These 3 are having 3 different mode of action, and which finally helps in the plant growth and soil health. Similarly, biocapsules, these are developed from ICAR, Indian Institute of Spices Research, Kozhikode, and these are successfully enhancing the shelf life of these PGPRs in this biocapsule formulation. And normally these application of these biocapsules is very easy, and they are more shelf life. And coming to biofertilizer efficiency enhancer, these are also all the new trends or new advances in these PGPRs related to soil health management. Similarly, there are several techniques for detecting of these PGPRs after they are applied into the soil. They can be detected by different techniques like fluorescent in situ hybridization, reverse transcriptase, polymerase chain reactions, terminal restriction fragment length polymorphism. So in this way, there are several advances related to these PGPRs which are helpful for the soil management and different crops growth. And finally, coming to the conclusion, these PGPRs main aim is to reduce the environmental pollution and improve the plant health, finally helping in the survival of the plant and reducing the environmental pollution. So this is a brief topic presented by me, sir. Thank you for giving me this opportunity, sir.
You, Dr. Kavya. I have two queries I have.
Yes, sir.
You have mentioned in one place Burkholderia and Klebsiella and all.
Yes, sir. Yes, sir.
If you the Burkholderia gives excellent result when you do in the in vitro assays. Same way Pseudomonas aeruginosa also.
Yes, sir.
Whether can we go with the mass production of these organisms commercially?
Sir, Pseudomonas aeruginosa, as of my knowledge, that can be gone for mass production, sir. But Burkholderia, I do not have a idea about that, sir.
Both are not permitted.
Okay, sir.
Both are not permitted. My second query, this topic is on the PGPR.
Yes, sir. Yes, sir.
The products, whether we have to follow fertilizer control order FCO Or should we go with the guidelines of the Central Insecticide Board, CIBRC registration committee? Which guideline we should follow for commercial production and selling?
FAO only, sir.
FAO or FCO?
FCO, sir, Fertilizer Control Order.
Yeah.
Because here we are not making any claims for the pest or disease. So it is
Yes,
only for
yes, sir.
promotion. That means
Yes, sir, yes, sir.
fertilizer control order.
Yes, sir, yes, sir.
Either, sir?
Yeah, this PGPRs, how best they can help in minimization of the pest, any idea?
Sir, they are having the capacity to produce hydrophores. They are having the capacity to produce some lytic enzymes, and they are able to grow fast with their inbuilt capacity, so they can compete with the other harmful organisms by this HCN production, hydrophore production. With these capabilities, they are able to suppress the pathogens and help the plant growth, sir.
A sort of, can you say, a sort of immunity boosting?
Yes, sir. Yes, sir. Yes, sir. Similarly, sir.
Okay, thank you.
You have mentioned as Serratia marcescens that produces exochitinase that inhibits the chitin synthesis in the insects.
Yes, sir.
Serratia marcescens
Harmful.
you have mentioned in one place.
Yes,
that
sir.
can be
Yes,
used
sir.
against insects.
Yes,
Right.
sir. Yes, sir.
Other queries? Thank you, Dr. Kavya.
Thank you, sir. Thank you, sir.
Go to next presentation. Anjali Kumari Jha. Ethnic vegetables as nutraceuticals. She is there. Dr. Tanuja, she is there. Anjali Kumari Jha.
This is not the topic.
No, this is bioactive compounds for disease prevention and management. It has been included here. Whether it is for plant disease or it is used in the previous session in the nutraceuticals. Yesterday, theme 2.
She presented yesterday.
Okay. By mistake, it has come here. Then next one is Dr. M. Ramaya, Professor and Head Entomology, Biosystematics of Leafhoppers in Vegetable Crops. Dr. Ramaya, sir, please. Size can be increased.
Shall I start, sir?
Yes, sir, please, sir.
Yeah, please.
Respected Chairman, sir, and Co-Chairman, and delegates and reports. So I'm Dr. Ramayam. Good morning, all of you. So here, this is my my student work. Now I am presenting. That is the biosystematics of leafhoppers of different vegetable crops. So regarding the introduction, all the leafhoppers are the major. These are the smaller insects. They have the wedge-shaped insects. So these have the brilliant color of the wings, and also the characteristic feature of these insects are the diagonal movement of the insects. They are ready for jumping. Always they sit on the leaf. They are ready for the jumping. So here we find the 2 rows of spines in the hind tibia. It is the specific characters of hoppers. These comes under the Cicadelidae family and order Hemiptera. So these are the excellent insects which can jump on the surface of the leaf. So and also have the wedge-shaped insect. It is a wedge-shaped insect. So regarding that is the introduction. Next, regarding the materials and methods, you have to collect these insects and preserve in the lab. The first you have to collect the insects and the drying of the insect and preserve for the next further study. Here first you have to take the China clay for the dissection of this insect and dissect the abdomen from the thorax. Then the abdomen you can kept in the cavity slides and kept for that 10% of KOH and can digest in the overnight. Then tomorrow we can collect the digestion, the abdomen, and collect these genitalia and kept it for the preservation and with the help of the glycerin. Tomorrow we can draw the diagrams of all the genitalia parts. So that is the stereozoom binocular microscope here they are using. So next you can see some of the insects. The genitalia are dissected by my student. That is the Amsecta albistregum, so Amsecta bigetula. So here specifically the idea is the major reason for the classification of the insect. Any insect is available newly, so you have to dissect the genitalia and describe the male genitalia. Here you can see the Amsecta bigetula. It is the tubular shaft of the Adiagus and slightly carved. So that is the specific character of this Amrasca bigetula. And also one more, we can see the Emposca kerri. It is the shaft. It is the Adiagus you can observe. The tubular knot is observed. So that is the specific. You can observe it is the bifurcated in the genitalia shaft. Next, we can go for the Bulcuta saltulella. So here we can identify this genitalia, the narrow curved and the elongated shaft. So here we can see the germphoid apically. So the gonophore is apically we can observe in the genitalia. So next we can go for the insect which you have the Histomonas ascites. It is present in the brinjal. So it is so strong here. The shaft is bifurcated with the border at the apex. The mesial margins are curved anteriorly. So that is the specific character of this Histomonas ascites adiagus. And next the Nepotis virescens. So you can see that it is the different shape we can see in the Nepotis virescens adiagus. So here we can see the 4 pairs of spines laterally we can observe. Under the pair of the lateral apophysis is also we can observe. This is specifically it is the virtual shape. It is the kitty shape, light kitty shape. It is up here. So next and further for the Dorolena rotundus. It is the excellent. We can observe the adiagus. It is the slender, very long, wider base, narrow abruptly, soft is slender, apex is pointed, conifer at the apical. So this is the specific character of this Dorolena rotundus. Next, the summary and conclusion. So you can see here the Amrasca bigetula and also the Emposca kerri and Bulcuta saltulella and Histomonas ascites, Nepotis virescens and Dorolena rotundus. Here we can see the Histomonas ascites is observed in the Nepotitics is observed in the turmeric and Dorolena rotundus it is observed in the brinjal. So remaining 2 species, that is the Dorolena rotundus and also the Nepotis virescens and Histomonas ascites. These are also generally degree stage we can study this Nepotis virescens in case of paddy, but it is now it is observed on these vegetable crops also. So specifically Histomonas ascites under the Nepotis virescens, it is a turmeric identification. It is the identification as my student work and Dorolena in the brinjal. Okay, sir.
Sridhar sir. Sir, Sridhar sir.
And at Ramya, this is one of the important group in insects. And in case of many of the horticulture crops, wherever these phytoplasmas are involved, these hoppers are recognized as vectors. In your collections, what are the important vectors of phytoplasmas observed other than Histomonas, one you have already mentioned? We are also seeing so much in China aster. The actual vector could not be identified. Whatever we gave, that Viratma told, they cannot be vectors. Means, is there any specific characters of vectoriferous hoppers in this Cicadelidae family?
Yes, sir. So many are there. Bemisia tabaci. So it is a vector, and also some of the Nepotis.
Only in this hoppers group.
Yes, sir. Nepotis virescens, it is observed earlier in the Gramineaceae family. It can transmit so many diseases, and also it is having the specific character. It can suck the juice of the plant, and the entire body is this. It is to the gut. The gut is entirely it is invaded. Then it is multiplied in the gut. and also the completely the life cycle is in the gut itself, and again it can through the oviposition itself, and also it can saliva of the particular hopper will transmit some of the viral particles. These are the small viral particles, and also the fungal particles. They can suck the juice, then it can transmit from the one plant to the another plant, sir.
Yeah, but though many, okay, that there is some specificity. Now, many of the hoppers, they are not transmitting. Some are transmitting very problematic things. So, yeah, thank you. That is the clarification I wanted. That's Sriram. Yeah, okay.
Thank you, sir.
Okay, sir. Thank you, sir. We'll go to next presentation. We have 4 more presentation. We'll try to complete in another 20 minutes. Dr. Sudharani, neem-based products as potent bioagents in pest management of vegetable crops.
Very good morning, sir. Am I audible, sir?
Audible, share the screen.
I request madam kindly share from your side, ma'am.
Yeah, share.
Meanwhile, a very good morning to one and all, respected chairman, co-chairman, and the organizing committee members. I am Dr. Sudharani, working as a senior scientist entomology in Acharya N.G. Ranga Agricultural University. So here with I am sharing, we have conducted a small research along with the RBK DRC team in many farmer fields. So what is the major objective here of our study is how best these neem-based products are used as potent bioagents in pest management of vegetable crops. So as we all know, vegetables are the primary sources of many nutrients and vitamins in our daily diet. But nowadays, if we see because of the commercialization and the developed competition among the vegetable growers, the residual effect on vegetables is increasing day by day, as we all know it. Maybe the basic reason for this residual effect is indiscriminate use of pesticides at over lethal dosages, or combination of 2 to 3 pesticides in a single spray. So at present, the need of R is to mitigate the health-related tribulations that are being caused by this residual effect in vegetables, and also to lower the environmental risks by these toxic pesticides. We have to shift to organic or biological based pest management tactics. So among these, one of the best initially for the earlier instars or the first and second instar larvae or the eggs of the pests, we are recommending the neem-based products. So we all know the neem-based products like neem oil, NSKE, the neem leaves, neem bark, and to the few extent, the neem flowers are also having a pivotal role in insect pest management of various vegetable crops. So they plays a key role as insecticides, fumigants, manures, and nowadays they are also using as urea coating agents, and also they improves the soil conditions. So the neem-based products can be used as a potent biocontrol agents, and as we all know, these are having active ingredient components like azadictin, nimbolidine, and selenin. So with respect to insecticidal properties, basically these neem products acts as antifidants, repellents, and repugnant agents, as they are inducing sterility by preventing oviposition in female insects and interrupting sperm production in male insects. And besides these neem-based products are also having compatibility with other insecticides, fungicides, nutrient foliar sprays. So these can be used with, these are compatible and hence they can be mixed, as the farmers are mostly interested to go for mixing of 2 to 3 chemicals. So we can recommend these commercial neem-based products also. And here I can also, we have conducted a small trail in the farmers field along with Raitu Bharosa Kendra of a district resource center of our Krishna district, Andhra Pradesh, where majorly we have concentrated on both tomato and turmeric crop. So initially, if we have sprayed the neem-based products and another organic module, so we could have recorded 72% reduction over control with respect to whitefly in tomato, and 58.58% with respect to fruit worm in tomato. Whereas in farmers' practice, they have recorded only 37% reduction with whitefly and 46% with regards to fruit worm in tomato. Whereas in turmeric nowadays, for the past 2 to 3 years, we are observing a severe damage of root grubs.
So here root crops farmers are spraying chemicals like anything. But if we have recommended to spray for neem-based pesticides in the nearby trees of where the adults are there, we could have reduced around 69% of damage caused due to the root grubs in turmeric, whereas in farmers' practices they have recorded only 49%. So with this, I can conclude with one or two points, because this is the trail that we have conducted in the farmers field. So I can accomplish the farmers, especially the vegetable growing farmers. They have to be encouraged to use these neem-based products, maybe through training come awareness programs or demonstrations. Demonstrations basically relating to how to prepare NSKE and how to prepare the spray fluid concentration, that neem-based spray fluid concentrations. We have to go for campaigns, and even our district collector and also the department officials are also supplying the required products on subsidy basis, especially these neem-based products. So with these few points, I would like to conclude that neem-based products are highly recommendable and suggestible to the vegetable crops, especially to the early stages of the pests, that is egg stages and first and second instar larvae, both for the sucking pests and also borers. So thank you for giving me this opportunity.
Thank you, Dr. Sudharani. Sir, Sridhar sir.
Yeah, Dr. Sudharani. Yeah, more emphasis is given for neem products. Can you share some of the products? What are the actual products used in your trial?
Sir, all these products.
At what doses? At what doses?
Sir, actually, all the products that we have used here are not the commercial based formulations, sir. We have, we have made the method demonstrations how to use these neem, especially we have used this neem oil, sir, and NSK. 5% neem oil and 5 ml per liter of water, neem oil and 5% is NSK were used in our trail. But however, all these bark, leaves, flowers, seed, and all these we have shown to the farmers how best we can use this. For root grubs, we have sprayed neem oil, sir.
Neem oil, if it is a pure neem oil, what is the percentage used?
Sir, 5%, sir.
No, 5% oil usage is too high.
Oil.
In any
No, sir, oil.
No,
sir,
if it is
oil
pure,
10,000.
is pure. No,
Yes, sir.
then that is a formulation. That is why I am using.
Right.
I am asking. There are, suppose if you are using neem oil as a pure commodity, if you want to use,
Yes,
generally we won't go more than 1%, 10 ml per liter. If it is pure, then you are telling it is from the neem natural products. If it is ppm, means again it is a oil based, maybe oil based formulation.
Yes, sir.
That's why I'm
Actually,
asking.
the formulations are being developed by Department of Agriculture, Krishna district. So, and we
Okay.
are commercializing through Raitu Bharosa Kendra, sir, and distributing to the farmers on subsidy basis, so as to decrease the pest load in the initial stages only, sir.
Yeah, that is true.
Yes,
Yes,
sir.
I am concerned with this doses, because if you see commercially also in among botanicals, neem products are the highest number of products which are registered with the CIBRC, not even any other insecticides, different formulations. In that respect, we should be careful with the dose and formulation what you are using. That 5% oil means it is, if it is a pure more,
Yes,
what is that
dilute,
10,000 ppm? 10,000 ppm of what? Can you elaborate? What is the 10,000 ppm?
Sir, actually, they are department was they are mentioning that neem oil 10,000 ppm at 5 ml per liter of water is their recommendation, sir.
No, when they are maintaining 10,000 ppm means it is 1%. What is
Yes,
that 1%?
1%, yes, sir.
Whether it is already it is oil is 1%, there is some azadirachtin in 1%. What is that? Just I am curious to know that. Okay,
Actually,
okay.
they are yet to be taken patents for commercialization. So maybe I will let you know, sir, because.
Okay. Please let me know. I'm interested to know about
Yes, sir. Yes, sir. Definitely, sir. I'll inform you. Sir.
Okay, thank you.
Thank you, sir. Thank you for your suggestion, sir. Thank you.
Thank you.
We'll go to next, Dr. Geetika.
Yes, sir.
चल रहा अभी कितना चल रहा है 2-3 सेशन
Sir,
है
am I
सर
audible?
चल रहा है पता नहीं चल रहा है इसका
You are audible.
टाइम नहीं बता
They are
दिया
forced to mute the others except the presenter.
प्रेजेंट करना है कहाँ हो रहा है ये आंध्र प्रदेश में
Can mute the others. You can present.
Geetika, are you sharing?
Am I audible, sir?
Yes.
Sir, I'm not getting your voice.
Yes, Geetika.
You are audible.
Geetika, you are audible. You can start.
Okay. Good morning, everyone. My name is G. Tirumal Geetika, MSc first year, Department of Entomology, College of Horticulture, Anantarajpeta. Today, my topic is biocontrol agents and its application methods. Coming to biopesticides, these are derived from microorganisms or natural products that control pests through different mechanisms of actions. Generally in India, neem-based products like neem seed kernel extract, neem oils, and microbes like Bacillus thuringiensis, NPV, and Trichoderma are widely used. These are available in both powder and liquid formulations. Coming to powder formulations, these are developed by mixing a binder, a dispersant, and a wetting agent. And coming to liquid formulations, these are of water emulsions or oil emulsions. Mostly preferred liquid formulations cause these have high cell count, prolonged shelf life over a year. And coming to the application methods, seed treatment, soil application, foliar spray, microbigation, congregation assay, and seed biopriming. Okay, coming to seed biopriming, this is a seed treatment that integrates the biological and physiological aspects of disease control. It involves coating of the seed with fungal or bacterial biocontrol agents. And these are incubated in warm and moist conditions just prior to radical emergence. And microbigation. Microbigation is application method which control weeds, soil pathogens, and soil insects. In this, biocontrol agents are transferred through the drip irrigation. And elaboration of this is conidial suspensions can pass through the drippers without causing clogging, regardless of their size. And some of the things are seed treatment of tomato seeds with the powder formulation of PGPR like Bacillus subtilis and Bacillus plumilis. These reduce the severity of tomato mosaic virus by 48%. And coming to seedling root dip of chili seedlings with liquid formulation of Trichoderma halcyanum. And these powder and liquid formulations of different colony forming units are used to treat the seeds for the diseases like tomato mosaic virus and seedling rot. In nursery management, seedling root dip method is highly efficient. And for the seed-borne diseases, seed treatment is efficient. And for soil-borne diseases, soil application method is very efficient. Thank you, sir.
Geetika, this is a compilation of the literature available, isn't it? Any work carried out by you?
Sir, I'm not getting you, sir. Can you please?
Because this all information is available,
Yes.
you have compiled and presenting it, isn't it?
Actually, single,
Or-
sir.
No, whether any part of your study you are presenting in this included.
No, sir. No, sir. I'm studying MSc first year, sir. I'm not done it any research on it.
Sridhar sir.
Yeah, okay. He have mentioned in some of the nowadays so many changes in the genera names are happening. One of the fungi what you mentioned is verticillium. Its name also has been changed to lecanicillium. Just note that one for
Yes.
future presentations. Okay.
Okay, sir. Okay, sir. Okay, sir. Sure. Thank you.
Okay. Then what are the effect of these formulations? There will be liquid formulations or solid formulations. In your idea, what is the advantage of this liquid as well as solid? Or both are same? Why different formulations?
Sir, liquid formulations, these have high cell count and prolonged shelf life, sir, over a year than compared to solid formulations.
That way. Okay, right.
And these liquid formulations, these have enhanced protection from environmental stresses. Coming to solid formulations, they may be accumulated in the soil for a meantime. Maybe, and coming to liquid formulations, these are like, I'm sorry, sir, I'm done.
Okay, yeah, in the track, you can track that one. Okay.
Okay, sir. Thank you.
Okay, thank you. Thank you. Thank you. Yeah.
Question, sir. Miss Sindhuja K. Studies on root knot nematode resistance in polyhouse tomato.
Yes, sir. Good morning, sir. Sir, am I audible, sir?
Very good. Share these slides.
Okay, sir. Sir, whether my poster is visible, sir? Sir?
Is visible. Please present.
Okay. Good morning, everyone. I'm Sindhuja K, pursuing Masters in Vegetable Science from College of Horticulture, Rajendra Nagar. As a part of my Master's program, I have worked on studies on root knot nematode resistance in polyhouse tomato cultivar grafted on turkey berry under the guidance of Dr. S. Mallesh in collaboration with NPPJ Regional Station, Hyderabad. As we know that nematodes became one of the major problem under protected cultivation, mainly in solanaceous vegetable crops, it is very difficult to manage once established in field. Some management practices through chemicals, crop rotations also not that much effective. So one effective eco-friendly technique is needed to overcome this problem is grafting. So with this background, we have formulated the null hypothesis to assess the root knot nematode resistance of polyhouse tomato cultivars grafted on turkey berry and to evaluate the grafted combination for growth, yield, quality of polyhouse tomato cultivars grafted on turkey berry against root knot nematode. So coming to material and method, first seed material. For grafting, we need rootstock and scion. For rootstock, Solanum torvum seeds were collected from NPPGR New Delhi, which was already partially resistant to nematodes. So Solanum torvum seeds has issue with germination. So we plan to conduct one independent experiment, that is effect of GA3 treatment on seed germination of turkey berry with different GA3 concentration from 0 to 2000 ppm at different durations of soaking from 6 to 24 hours. So by which rootstock were raised. After 45 days of rootstock, scion seeds were sown. 3 different scion materials were taken: Arka Samrat, Saho, SVTD 8323. So when it reached 30 days old, we done grafting by following cleft grafting method using polythene cover. So this grafted seedlings are used for 2 experiments. So first experiment was to assess root knot nematode resistance parameter, which was conducted under fully automated polyhouse with CRD design. So coming to second experiment is conducted at naturally ventilated polyhouse to assess growth, yield, quality and nematode resistance under FRBD with 2 factors: grafted combination and presence or absence of nematodes. Let's move on to the resultant discussion. From experiment 1, we have proved that GA3 2000 ppm at 24 hours of soaking significantly best compared to others. Here we can see the difference. 250 ppm at 24 hours recorded less percentage of germination, 70%, and also slow growth of the seedlings. So from experiment 2, we have studied the resistance against nematodes. So grafted seedlings were found growing better without any foliar symptoms. In case of non-grafted seedlings, it's clearly shown that high difference with foliar symptoms. So in experiment 3, grafted plants were found significantly better in growth, yield, quality under presence of nematode. Here we can see the clear-cut difference. So grafted plants were least infected with nematodes, that is very less than 10 number of galls. In case of non-grafted plants, it had uncountable number of galls and more different in root system also. So coming to the conclusion, we determined the optimum GA3 concentration and duration of soaking of turkey berry seeds for getting better germination, we found the resistance of grafted plants against root knot nematode, and we determined that the compatibility level of tomato varieties grafted on turkey berry. Thank you, sir. Thank you, everyone.
Presentation. Sindhuja, I have 2 queries.
Yes, sir.
What was the stage of the nematode used for inoculation? Root knot infector juveniles we use. Na? Which
Yes, sir.
stage of IGs you used for inoculation?
Sir, actually, already infested nematode 6 soil we used, sir.
What was the population then?
Sir, for 200 cc soil, we used 200 nematodes.
Yes, sir.
Yeah, this one for Solanum thoramum. First thing is you have standardized GA concentration.
Yes, sir.
Even after GA concentration, it is taking around 3 weeks for its germination.
No, sir. No, sir.
At what age of the plant grafting you have taken up?
75 days for root structure, 30 days for scion, sir.
75 days minimum required. What is the general girth of that stem approximately?
Sir, general girth, sir.
Because
Yes,
for
sir.
grafting we need minimum.
Yes, sir. Yes, sir. 2 centimeters.
2 centimeters.
No, sir. No, sir. Sir. 20. Sir,
is too low.
actually in centimeter,
is too high.
No,
too high.
1.5, sir. 1.5 centimeters, sir, actually.
0.5 centimeter is too big. Even matured brinjal will have that much. It's the generally pencil thickness, that is the recommended thing for grafting purpose. Whether it is reaching that level or not, that is my interest of this knowing.
Okay, sir. But
who has done grafting? Yourself or somebody
myself.
help?
Myself, sir.
Yourself. What is the success rate? How many you did and how many success grafts you got?
88% success I got, sir.
What conditions you gave to these plants? What humidity and what temperature, or just left in the outside?
Just left in
Generally,
outside
grafting
only,
needs
sir.
some specific conditions of RH for better.
Yeah, sir. But I did in shade condition only, sir. I didn't maintain any mist chambers.
Normal. Okay.
Sir.
Okay. Okay. Right.
Thank you, sir.
Okay. Thanks.
So we are going for the last presentation. Dr. Ruth Madam, Professor and Head, Department of Plant Pathology, on prevalent disease of money in Andhra Pradesh. Ruth Madam is there?
Yes, sir. Sir, madam is here.
Good morning, sir.
Good morning, madam.
Good morning, sir.
Good morning, madam.
Sir, my poster presentation on prevalent diseases and challenges in onion crop in Andhra Pradesh. Onion is one of the most important commercial vegetable crop grown in India, and Kurnool is the leading district in production and area of this crop, followed by Anantapur and Kadapa districts.
The major onion producing states are Maharashtra, followed by Karnataka, and other states also this crop is cultivating. And there is a lot of demand of Indian onion in the world. And also India is the second largest onion growing country in the world. And it is famous for their pungency, and they are available throughout the year. And the major varieties are Agrifound dark red, Agrifound light red, and Agrifound white, and other NHRDF varieties are cultivating in Kurnool district. The first objective is to know the prevalent diseases and challenges in onion crop in Andhra Pradesh, and conducted field experiment and conducted rules to monitor the diseases occurring in Kurnool and Kadapa districts. And in results, onion, in case of onion, the purple leaf blotch, it is a major disease. The percent disease index is 36.5 and damping of in nursery stage, and it's not the bacillus of diseases and the stem elongation, and mosquito, it is one of the problem. And the tricks are the major production, major problems identified in different methods. And the more is in Kharif and distant case of elongation and please observe the leaf blight disease. And also in survey, there is some lodging and overgrowth at the root portion was observed due to the high doses of nitrogen fertilizers, and which leads to the problem of mishap and bulbs. And while digging of bulbs also, it is a problem in black soils. And black soils are not suitable for onion cultivation also. And the premature bolting, it is one of the problem in Kharif season. And also there is a post-harvest problems are also observed in case of onion cultivation. The major problems are improper grading and selection of the quality bulbs, and also some lack of storage structures also observed in case of onion cultivation. And generally, coming to the conclusions, the major challenges are availability of spurious seed and the lack of availability of improved varieties, and also lodging and overgrowth of root portion or recalculation in some varieties, and extra growth at root zone portion that is not suitable for marketing also, and due to high extra growth of the at the root portion, it is maybe due to high quantity of nitrogenous fertilizers, and there is some thrips problem, and the storage structures also it is a problem, and also price fluctuations problems are also there in case of onion cultivation. So the farmers are suffering from this for this problem on price fluctuations, and and also there is storage structures availability is there, but the farmers are not accepting these storage structures, and these are the major problems in onion cultivation. Thank you, sir.
Thank you. Madam, I have just two queries. Do you have stemphyllium incident severity also in your place? Other
Yes,
than
sir.
blotch, stemphyllium, between the onion blotch and the stemphyllium, which one is alternaria and stemphyllium, which is very severe in your place?
Yes, sir. Alternaria pure it is a major problem, sir, and also some Coltotrichum problem is it is also there in some mandals of Kurnool district.
I'm asking about stemphyllium, madam.
Stemphyllium, it is not there, sir. Stemphyllium, it is not observed in our areas.
What about the other bulb rot and all, madam? Because a waterlogging condition, generally bulb rot is coming.
Yes, sir. Waterlogging conditions at the time of storage, at the time of harvesting, it is a problem. But the farmers are not following the water management at the time of harvesting stage, sir.
What are the species of Coltotrichum you are getting now, madam?
Sir,
Predominantly,
Coltotrichum,
which
it is-
species?
the species are not observing, sir. Only genus is identified, sir.
Sridhar, sir?
It is maybe Coltotrichum gliosporidis, or it is not confirmed till now.
So,
In
madam,
our
in Kurnool area, whether farmers are following that raised bed system or they have some problem using the technology?
Sir?
Raised bed system of
No, sir. Some farmers are following, sir, but majority of the farmers broadcasting that onion seed, and after that they.
Okay. Is there any incidence of mites along with thrips observed in that area? You have emphasized thrips, but not mites. That generally in many places, this mites also a problem. Whether it is observed?
Sir, there is no mite problem. Actually, thrips is a major problem in onion in this area, sir.
Okay.
Thrips and only mites are not observed, sir.
Yeah, in rainy season, what is the major problem? Major disease? Because that is the factor which will dictate the prices after the harvest. What diseases are posing a problem after the harvest, particularly during rainy season?
Rainy season, there is a purple leaf blotch. It is a major problem, sir. After harvesting the problem, it is a basal stem, basal bulb rot. It is a problem, sir. And some only purple blotch. It is a major problem, sir. Followed by this leaf blight, sir.
In Kurnool, any storage structures are there? Post-harvest storage structures,
Yes,
provision
sir,
is there?
Yes, sir. Some storage structures are developed. That is sanctioned by EPEDA and some government of Andhra Pradesh, sir. But farmers are not utilized properly.
Because that will give a lot of scope for this buffer in absorbing this cost. Very low price means they need not to sell. They can store and sell it after some time. That's how they have to use. This year particularly very pathetic price-wise. Even they didn't get, even in Maharashtra, 3-4 rupees also they sold. That's
Yes, sir.
yeah.
Yes, sir.
Okay. Right.
Yes, sir. And Ballari Red, it is a local variety, sir. Farmers are using that, cultivating that Ballari Red only, sir.
Ballari Red, okay.
Ballari Red, sir. Followed by Agrifon Dark Reds.
Okay.
Thank you, sir. Thank you for giving this opportunity.
Okay. Thank you, madam.
With this, we are coming to end. Sir, your remarks you want to make, sir? Sridhar, sir?
Yeah. No, you will be making in detail now. Okay. We appreciate all the presentations. We had presentations from pathology, entomology, nematology as well. We also could learn. It is a cross talk. This is the purpose of the seminars, is to exchange these ideas. I may be knowing some present ones I will be learning. And this is how the scientific communities to get benefited. Many of the students, their presentations are also good, and many are on track. And what we try to emphasize is what the questions or clarification we asked is for the betterment only. When we also started similar type of things, helped us to develop. That way, you have to take all these questions, cross questions, clarifications in positive way. With this, I congratulate all the presenters for your nice work, and hopefully we'll come out with good recommendations for this conference. Over to Dr. Sriram. Yeah.
Thank you, sir. As Sridhar sir told, for us it is opportunity to learn what is happening in the other places also. And especially the recent topics taken by the students will help us to understand what is happening in the other places. And almost all made a very nice presentation. And for students, I want to add one point. Most of the students, when they presented, they put their name only. They could have added their guide name and others who have helped in that paper. I saw some of the students' presentation, only the name of the student was there. Otherwise, everybody has made a very good attempt, and thank the organizers for efficiently managing the program and giving us the opportunity. I once again thank the YSR Horticulture University for giving this opportunity, especially for Dr. Gopal, who is the Associate Dean. I sincerely thank the rapporteurs, Dr. Arunodayam, Dr. Nischala, and Dr. Dinesh, and Dr. Tanuja specifically. And she is in touch with us from yesterday and nicely coordinating. On behalf of all of you, I thank the organizers. Thank you very much.
Yeah, thanks to the organizers from my side also, whomever you covered. For everyone, I too thank you personally. Right. Yeah, we are almost in time. Only half an hour. That is because of yesterday's delay. Otherwise, it is consider it is on time. Hopefully, Then now, Dr. Tanuja, you can take over.
Yes, sir. On behalf of the organizing team, it's my privilege to propose a vote of thanks on this occasion. I feel very deeply gratified and thankful to the chairman, Dr. Sriram sir, and co-chairman Sridhar sir, for being with us for the last 2 days and the smooth conducting of the program and enlightening the session with giving their suggestions and giving recommendation in the field of research and improving their respective field of research. And I am also thankful to the invited speakers, Dr. Sridhar sir and Dr. Ruth madam, for giving light on the latest development in the field of pest and disease management. I'm also thankful to all the delegates for sparing their valuable time, but also for the intellectual deliberations during the session. I'm short of running of the words to express my humble thanks to all the participants and for sticking on their time. I would also like to thank the efforts made by the rapporteurs and the students for the smooth conducting of the session. Thank you, one another. Thank you, sir. Thank you.
What is next? We'll close this session. Okay, right.
Thank you, everybody.
Thank you. Thank you, everyone. We'll meet. Yes.